All right, today we are diving deep into one of the most amazing fundamental processes in all of biology. It's called translation. And really, it's this incredible molecular machine that's humming away inside every single one of your cells, right now, reading your genetic code and building the very proteins that make you, well, you.
I mean, think about it for a second. It's a pretty mind-blowing question, right? If your DNA is basically the master software for your entire body, how in the world does that line of code become a living, breathing thing, a physical reality? So, yeah, let's totally unpack that. So, to understand this, we've got to talk about what's known as the central dogma of biology. It sounds kind of intimidating, but it's really just the roadmap for how genetic information flows. And translation, the process we're zeroing in on today, is the absolutely crucial final act in this play. It's where the code finally becomes creation.
Okay, so here's how it works. Think of it like this. Your DNA, that's the master blueprint. It's priceless, so it's kept safe in the head office. You'd never take those original plans to a messy construction site, right? No way. So instead, you make a temporary working copy, and that's RNA. This RNA copy then gets sent out to the factory floor to be translated into the real functional product, a protein. DNA to RNA to protein, that's the flow.
But here's the thing. Before scientists could even begin to understand this factory process, they had a huge problem to solve first. They had to crack life's secret language. And back in the 1950s, a physicist named George Gamow tackled this, not in a lab, but as a pure logic puzzle, which is just amazing when you think about it.
So the RNA language is ridiculously simple. It has just 4 chemical letters in its alphabet, A, U, G, and C. That's it. Just 4 characters to write the entire instruction manual for life.
But here's the challenge, right? The whole point is to build proteins, and proteins are made from 20 different building blocks, these things called amino acids. So here's the puzzle Gamow was looking at. How do you use a tiny 4-letter alphabet to create enough unique words to stand for all 20 of those amino acids?
Well, the answer turned out to be just simple math. Let's walk through it. What if each genetic word, which we call a codon, was just 1 letter long? Well, you'd only have 4 words, not nearly enough for 20 amino acids. Okay, what about a 2-letter code? That gives you 4 times 4, or 16 possible combinations. We're getting closer, but still, nope, not enough. So what about a 3-letter code? That's 4 times 4 times 4, which gives you 64 possible combinations. Bingo, that's more than enough. This simple logic was how they figured out the genetic code had to be read in groups of 3, a triplet code.
Okay, so now that we've cracked the code, let's meet the key players on the assembly line. I really want you to keep this image in your head. We're talking about a super high-tech factory that's running inside every one of your cells.
First up, we've got messenger RNA, or mRNA for short. This is our blueprint. It's not the original, precious DNA, remember? It's a temporary, kind of disposable copy of just one gene's instructions. And its whole job is to carry that blueprint from the protected central office, the nucleus, out to the busy, noisy factory floor. Next, say hello to the transfer RNA, or tRNA. And this guy is our specialized delivery truck. It's incredibly clever. See, one end of the truck is designed to read a specific 3-letter codon on the mRNA blueprint. And the other end, it's always carrying the one specific amino acid that matches that codon. So it's both a translator and a delivery truck all in one. Super efficient. And finally, we have the factory itself, the ribosome. This thing is the real workhorse. It's a massive molecular machine made of 2 big pieces, and its job is to clamp down onto that mRNA blueprint and act as the workbench for the whole construction project. This is where all the magic happens. Now, if we could zoom in on this factory, we'd see it has 3 key docking stations. Just think APE. The A site is the arrival dock, where new tRNA delivery trucks pull in. The P site is the processing station, where the growing protein chain is held. And the E site is the exit dock. That's where the empty trucks leave to go pick up a new load. Arrival, processing, exit. Simple as that. All right, we've met the crew, we know the layout of the factory. Let's put this assembly line into motion and see how it works. The whole show runs in 3 main acts: initiation, elongation, and termination. Let's break them down. Okay, phase 1, initiation. This is the setup. First, the small part of the ribosome factory latches onto the mRNA blueprint. It then slides along until it finds the specific 3-letter sequence that says start here.
That's the codon AUG. Once it finds it, the very first tRNA truck carrying the amino acid methionine locks into place. Then, bam, the large part of the ribosome clamps down on top of everything. The factory is officially open for business. Now we hit the main cycle, which is called elongation. This is where the real building happens. A brand new tRNA truck carrying the correct amino acid for the next codon lands in that open arrival dock, the A site. Instantly, the ribosome does its thing, creating a strong chemical bond that links this new amino acid to the growing chain. And then, the whole ribosome machine lurches forward exactly one codon down the blueprint. And this sets up a perfect repetitive cycle. You see, when the ribosome shifts, the now empty tRNA that was just holding the chain gets booted out of the exit dock. The tRNA that's now holding the longer protein chain moves into the processing spot, which leaves the arrival dock wide open for the next delivery truck. So, a new tRNA arrives, the chain gets one piece longer, the ribosome shifts, an old tRNA leaves, and this just repeats over and over and over again, adding amino acids at an absolutely incredible speed. And finally, we get to termination. The ribosome keeps chugging along the blueprint until it hits a stop codon. Now, there's no tRNA delivery truck for this code. Instead, a special protein called a release factor slots into the arrival dock. This acts like a big red stop button. It triggers a kind of molecular scissor that snips the finished protein chain, setting it free, and with the job done, the whole factory just falls apart, disassembles, ready to be used on the next blueprint that comes along. So that's the whole process, from start to finish. But let's zoom out for a second. Why is this tiny factory such a big deal? Well, its efficiency is just staggering. A single one of these ribosomes can add up to 20 amino acids to a growing chain every single second. I mean, that's just mind-bogglingly fast. And the output of all these factories is the foundation for pretty much everything your body is and does. And if you think that's efficient, it gets even better. The cell doesn't just use one factory on one blueprint at a time. Oh no, that's not good enough. Instead, you can have a whole bunch of ribosomes all working on the exact same mRNA blueprint at the same time. It's like a convoy of factories all chugging down a single assembly line. This whole setup is called a polysome, and it is mass production at the molecular level, just churning out insane quantities of whatever protein is needed and fast. So what is all this furious work producing? What do these proteins actually do? Well, basically everything. They are the building blocks that give you physical structure. Think collagen in your skin or keratin in your hair. They are the functional workers, like the enzymes that digest your lunch. And they're the messengers, the hormones and receptors that let your cells talk to each other. They're the whole show. This whole system, this incredibly elegant and precise factory, is what makes life as we know it possible. But here's the catch. The factory is only as good as the blueprint it's given to read. Which leads to a final, really important question. What happens when there's a typo in that code? What happens when the assembly line is given the wrong instructions? Well, that's where things can go wrong. That is the molecular basis for so many genetic diseases. And that's something to think about. Thanks so much for diving into this with me.